We're going to talk about the DNA molecule and its structure and DNA replication. So we have a DNA molecule here. It is double-stranded. We have a strand up above and a strand down below. And they are joined together by hydrogen bonds in the middle. Hydrogen bonds are very weak, and these will be broken later in DNA replication. So DNA is usually in a double helix. That means it's twisted. Right now it's just laid out thin like here and flat. So to get an idea, each one of DNA molecule here is made of tons, millions of nucleotides. And so a nucleotide contains three parts: a nitrogenous base, a sugar, and a phosphate group. Now we can break those down like we have in this picture here. We see our nitrogenous base, our deoxyribose, which is a sugar, and our phosphate group. Now remember, if it ends with OSE, that's a good idea that we have a sugar there. So DNA is also read in a 5' to 3' direction. We see that direction in our molecule here. Shows us the arrows, 5 to 3. But what does that mean? Well, we're talking about the carbons on the sugar. There are 5 carbons in the sugar, and depending on which carbon is read first or encountered first with what is known as the DNA polymerase, an enzyme that will put together our copies, that dictates the 5 to 3 direction. So to do that, we find the nitrogenous base and we look for that first carbon. That's carbon 1. And we're going to go in a clockwise fashion here. And there's 2, and there's 3, and there's 4, and 5. So we have an oxygen up here. So again, 1, 2, 3, 4, 5. In this next diagram here, we can see again, we have our 5' carbon right here of our deoxyribose. And down here, there is no C, but in science it's understood that if there's no C here in a ring like this, it's carbon. So this is actually 1, 2, 3, 4, and 5. And since the 5 carbon is encountered before the 3rd carbon as it's being read, then this is the 5' end, and down below is the 3' end. On the opposite side, we have the DNA strand going in the opposite direction. It is anti-parallel. In this case, everything's upside down and flipped. And here's the 5 carbon here. How do I know that? Well, again, I start my nitrogenous base, and I go 1, 2, 3, 4, 5. And so the 3' carbon is encountered before the 5th one here. It's on top. But again, it's not read in that direction. Our arrow shows here that it's still read in a 5 to 3' direction. So when DNA polymerase reads our DNA molecule for replication, it's going to be reading each strand in a different direction. So let's take a look at DNA replication. What we have here is helicase. Helicase is a protein, is an enzyme, and its job is to unzip or break apart the hydrogen bonds between each nitrogenous base of these nucleotides here. For instance, as I move it through, we'll start the first one with our fingers here, and we can see that this wedge here helps unzip it, and it breaks apart the hydrogen bonds. And we keep moving it through, and it's doing a fantastic job here. And what we want to do is we want to unzip this so then that we can then read it and replicate each of these new single strands that are coming apart here due to this helicase. So we have now started the unwinding process here. We're not finished. In fact, now we're going to bring in 2 more enzymes. A DNA polymerase right here, and a DNA polymerase, another one, down below. Now, we have a leading strand and a lagging strand. So the leading strand is the one on top, and the lagging strand is the one down below. This will up here be a continuous construction of a new DNA double-stranded molecule. Down below, it'll be considered discontinuous, and I'll show you why here in a moment. But first, we're going to focus on our continuous leading strand and how it's all put together. So, like we said before, this is all read in a 5 to 3 direction here. We'll follow our arrows here. And so what I'm going to do is with this DNA polymerase, there's an active site. The active site means where there's going to be the bonding of the free nucleotides. Ooh, free. And they're going to be brought in by the DNA polymerase, and they're going to bind with this strand of the parent DNA molecule. So I'm going to go ahead and put a T here with the A because adenine always pairs with thymine. And then I'm going to move it on down. And now we have cytosine, which will pair with guanine. And we keep moving. We have another cytosine with guanine. And now we have a guanine with a cytosine. C with G, G with C every time, A with T, T with A every time, and DNA replication, that is. Things get a little bit different when we get into transcription, but that's for another time. And as we can see here, we're creating one continuous new DNA molecule that is a copy of the parent molecule. And we have conserved some of that parent molecule into this new copy. So this is considered to be semi-conservative because we have conserved some of it into this new copy, and down below we'll be doing the same thing. So let's take a look at down below now. So this is our lagging strand, and our arrows dictate how everything's being read. We saw it as being read right here, right, 5 to 3. And then down here, the reading is going in that direction. So this is the direction that the DNA polymerase is going to move. I'm going to go ahead and place that DNA polymerase in there like so. And we're going to go ahead and start putting in our free nucleotides. Same process, C with G, G with C. And notice how the DNA moves. We're going to end up hitting a bump here. So what happens is this polymerase will come off, and it will wait until more DNA has been fed through. And we would continue the process up here as well too. And then it comes in and reattaches. And we can see it keeps going, all right? Keeps moving in the direction that is showing here. Keeps going, and then it hits a bump up here, an end point where it can't go any further. So at that point, it detaches, and we go ahead, and there would be another enzyme that would put this all together here, these final pieces. These two fragments, right? And so we see is a discontinuous process. Again, it has to wait until more comes out, and then it comes in, and it continues the process again until it again hits where there's already a full copy made. So again, this is the continuous section up here. This is the discontinuous section down here, the leading and the lagging.